Before I forget, I'll start a recording. I do enjoy making recordings and having a YouTube channel. It's hidden, but still, it's a lot of fun. Sorry for the grayish things. I'll I'll make them disappear. At the beginning of a PowerPoint, it's usually like that. Okay, please stop it. There we are. There's it. Yeah. This is going to be topic 11, and we have 42 main topics, but obviously a lot of these ones are revision, especially in chemistry. So in certain weeks we might take two topics in biology. Bottom line is I would want to finish everything by the end of February, the latest.
Yes.
And if possible, to have more and more practice sessions. I'm going to make you talk, and this sounds a little bit threatening, but it's not a threat. Why? In order to be able to convince these buggers in the entrance exam. Now, obviously, you've already learned quite a bit about DNA in your regular classes.
Yes.
I'm certain about its organization and whatnot. Um, my topics are set for people who haven't really studied any chemistry whatsoever or any biology whatsoever. As you can see, the way I'm trying to teach you things is from the perspective of physiology, because out of physiology grows internal medicine, diagnostics, a number of other things that are going to be your bread and butter as a medical professional. And once you understand and see the whole body as a whole thing, well, nobody can challenge your opinion because you'll be able to defend it. Now, we're going to talk a tad bit about nucleotide chemistry in a nutshell. It's a good idea to know a lot of these structures, but oftentimes they won't expect you to know it necessarily. But if you do, it bumps up your score and it bumps up the impression you make. I wasn't at all because recently, in the past 3 years, there was only one evaluator in the entrance exam for the international line of study. And it seems a little bit weird because for the Hungarian kids, we have a president of a committee who's usually a full professor. We have an associate professor next to the full professor and the student representative to make sure that the students' rights have been respected.
Mhm.
So I did make a bit of noise about this within Semmelweis as well. And of course, I would very much like if nearby medical schools would work together. Medical schools in Hungary and also where Hungarians live outside of current day Hungary, we should work together. Why? Because history shows that when we work together, it doesn't matter what language locals speak or what language locals don't speak. Everyone speaks English, let's face it, and
Mhm.
everyone wants scientific development. We should make it into a whole because when we agree and when we work together, we get things done. When we mess with one another, everyone becomes poorer and everyone is worse off.
Mhm.
That's what history taught me, but I'm not sure if everyone else was listening. Some people believe, you know what we need? We need to suffer more because that's always beneficial, isn't it?
Of course.
You have to tell entire generations not to touch the stove because it's hot. So everyone has the same wound on the same hand at the Yes, it's this kind of stupidity we're up against, my dear. Indeed. The double helix structure has been discovered in the 1950s. Obviously, nowadays, people do like to twist things in a way to fuel an agenda. Bottom line is, great scientists are not always great people, or great artists are not always great people, or great revolutionaries are not great people. But yeah, let's face it. One of my friends got fired from a job because he said Kossuth was a piece of shit. He was. He was. He was a lawyer, and the first order of business is what he did is he took the inheritance of orphans. Then they forced him to pay it back. That's why he was in jail. Yeah, our
Oh.
great hero. So I'm not a huge fan of these kind of movements or football, whatnot. I mean, I love people in general, and I'm just not inclined to blindly believe in politicians, and I advise everyone else to do the same. We should find certain character traits that obviously tell people apart, and you can easily find that. You just observe how people act with people they can expect nothing from, and if they're being polite, from the cleaning lady to the CEO to everybody, that means that yeah, that person has character. Yeah, that's what Granddad taught me. Indirectly, because I didn't know him, I inherited his friends. Then we're going to talk about chromosome organization. Why do we need to organize DNA? Imagine this. I have 2 meters of DNA per cell. How can I make that small and fit into a nucleus?
Uh, by the 3D confirmation.
Yes, and also, what do I do with it?
Wrap it around histones.
Perfect. Am I even teaching you anything new here? Because it's so amazing that you go through these and there's nothing new. I advise you to mainly follow Mr. Solomon's book. Thank you kindly. I will go over it after class.
This
I
is
will get back to you in the evening.
of course.
Go ahead.
No, I just it was up the file was uploading until now, so that's why it
Oh
came.
yeah, yeah, yeah. Recently, Telegram is much more controlled. It's what I've observed. The newly registered users are being asked for a 500 forint fee. I think they're trying to make it safer. I don't
Uh-huh.
know, because there were some bad people out there using it for bad things. Um, my Syrian kids are honest, quite straightforward. I used to teach at an international high school. I left because of reasons, but I really love these boys. I mean, imagine these huge kids who are like giant toddlers in anatomy and in thinking as well. They lifted me up and started tossing me from one another, and at the time I was 115 kilos. So, yeah, yeah, it wasn't an easy feat. And they asked me, why am I on Telegram? Am I soliciting adult content and prostitution, or am I working illegally, or am I a terrorist? I told them one doesn't exclude the other. How about all three? I love them dearly. They're such amazing kids. They speak like 3 or 4 languages and super kind-hearted. Imagine a huge baby with a couple of big people toys and sometimes doing stupid things because of the lack of proper parental adult supervision. Because there's a lot of them. There's like 4 or 5 siblings, so it's difficult to mentor all of them.
Mhm.
Yeah, I kind of get it, but I don't get it. But if you lead their character in the right direction, you make them do sports, you make them part of a team, they're going to turn into amazing young people, and after a while they're not going to be as Muslim, and after a while they just don't really care. They'll just have fun and just be one of us as everybody else. I mean, there's
Mhm.
nothing wrong with being this or that. Just don't be a bad person about it, I guess. Also, copying your DNA is very important. I mean, um, there's an author, a renowned atheist by the name of Richard Dawkins. He was very popular and quite present in the media while he was bashing Christianity. Now that he started bashing other religions, not just Christianity, all of a sudden he fell out of favor. To me, he's an interesting character. I'm not saying I agree or disagree with him. I find him interesting. And you can listen to someone, you can understand someone without becoming that person.
Mhm.
Just a suggestion. And yeah, he said that genes are rather selfish and they want to copy themselves. That is why every living being is trying to have as many offspring as possible. We've learned about 2 types of strategies in nature. Some have many offsprings but don't particularly take care of them. Others have fewer, but they put everything in those few offspring, whom are going to be successful through proper nurture. We have our own strategies regarding that as well. Some people who want to be funny, they say that about people as well, but it's not true. People have a limited lifespan and limited resources. What do we need to know at the end? Explain nucleotide chemistry and the formation of phosphodiester bonds, because it's a polyester basically. Describe the structure and properties of the DNA double helix. Perhaps they will want us to go into experiments, but usually I don't ask for such things because I think it's irrelevant.
Hmm.
Relate chromatin organization to chromosome structure and gene regulation. Explain the molecular machinery of DNA replication and uh, how is it achieved in detail? Obviously, it doesn't have to be that detailed at this point, but I'm going to go up to uni level with this because the more you know, the better impression you're going to create.
Hmm.
Oftentimes, people don't know what an IB is.
Yes.
I had a girl come to the Faculty of Health Sciences for Dietetics, and she said she has an IB diploma, and I was the only one who was being impressed. Everyone else was like, "What is that?" Like, dude, it's like being told that caviar is basically a type of fish egg that's really good. Um, seriously, you don't know what an IB is. Did you know that Kim Jong-un actually finished an IB?
Yes.
You can be a dictator at this point. I wish you were, actually, because it would probably put the world in a better direction, I guess. Yeah. I had South Korean Kims in my classes, and one of the boys asked me, "Sir, how should we call you, Mister, Doctor?" I'm like, "You should call me Daddy." He started laughing, and he's like, "No, that's not appropriate. I'm gonna call you Pops." And since then, he's been calling me Pops. I think that's for the best. Such a great kid. He made it immediately to uni after graduating McDaniel, and yeah, I'm rather fond of him. Now, depending on what's what, obviously you have a pentose. In the case of DNA, the sugar part, the pentose is deoxyribose. As you can see, it's missing. First time I gave my son short legged pants, he was like, the rest is missing. Where is it? He was surprised. What the heck is this? And I'm like, no, that's good. That's how it's supposed to be because it's hot outside. Well, in this case, the OH group is missing. In the case of deoxyribose, it's necessary. You shall see later on why.
Hmm.
And of course, thymine is characteristic mainly as a base, main characteristic of DNA. Uracil is mainly characteristic of RNA.
Yes.
This being biology, does this mean this occurs in every case?
No.
No, but we're going to ignore the ones that do. Because we're not geneticists and research biologists in this field. You can say mostly and you can be right. Oftentimes in high school books, they say always and never. In biology, we should be very careful on how we evaluate living beings because in the 90s and in the early 2000s they would say, oh, certain organisms are more evolved than others. They're not. They might be more ancient and haven't changed since then, but the level of evolution basically means that are you doing well in your surroundings? Yes. Are you thriving in your surroundings? Yes. Congratulations, you reached the pinnacle of your evolution. Yep, that's the bottom line. That's the next concept. And what about chemical stability? DNA is particularly stable because DNA is always inside the nucleus. RNA is less stable because a lot of RNAs are outside the nucleus. They're in the cytoplasm. mRNA, for example, is the template for protein synthesis. Yes. So structure and stability are very important because that is how we are able to encode genetic data. And it needs to have proper fidelity so the appropriate traits are going to be inherited.
Hmm.
Um, is DNA the only thing responsible for inheritance?
No.
No, not at all. A number of other things can also be inherited. Certain proteins may influence which genes are going to be active or inactive. However, the main template is still going to be DNA, but through development, a fetus may have issues that otherwise you wouldn't even imagine. For example, a couple of years ago, we found out that certain cosmetics may contain compounds that may have bad effects on a fetus. Oftentimes, we call them teratogenic effects if they cause developmental issues. I've known a boy who was born with his foramen ovale in his heart open, and he had to undergo several surgeries. And he lived up to the age of 18. He collapsed on his own prom and died there, and both of his siblings, um, medical doctors, they tried to revive him. He was in a coma for longer periods of time. Um, we pulled some strings. They got him an artificial heart in Frankfurt, Germany, but he was declared brain dead, so
Mm.
they pulled the plug. The only thing I regret is not being able to revive him because he was such a great kid.
Hmm.
He's the third child of my friend and colleague, and she's an amazing person. And it's the worst case scenario you can see is a mother having to mourn a child. It's
Hmm.
I mean, don't get me wrong, I think it's amazing that they got these 18 years as well. I don't want to be evil. I mean, I don't want to say that, oh, yeah, why so soon? Because, yeah, obviously, but a mother always wants that child to thrive and to outlive them, definitely. Because if it were, let's say, 20 years ago, the child would have died. If he was born 20 years earlier, would have died after birth, and that would have been it. But they managed to save him, and I don't know who made a mistake at this point. It's just a very sad story. And what can we take away from it? When we decide that we want children, we have to make sure that we keep our body in absolute health 1 year before actually deciding, and throughout development of the child, everyone has to be as healthy as possible.
Hmm.
Oftentimes, this is not being told to teenagers. Oftentimes, we ourselves underwent self-destructive behaviors because it seemed fun at the time. It turns out it can have lasting effects, not just on us, because on us, we might not even care that much, but on our unborn children, and nobody wants their child to be sick.
Yes.
So, when educating others, never forget to say these, especially with children and with teenagers, because your generation is far more responsible than mine was. I mean, boys still do stupid things, but they do it in a way that they don't harm each other or themselves, and that's amazing. DNA can be used as long-term storage. They have started experimenting storing data in DNA. And
Hmm.
I would very much like it, but we have micro SD cards that are 2 terabytes. 2 terabytes, that is an insane amount of data. I remember when all of my life would be able to fit on a 1 gigabyte blue flash drive. And me having that blue flash drive was absolutely high tech at the time. I mean, I had to carry a CD to install it on the computer where I wanted to use it. That is how outdated the technology of the time was. But then more and more computers started recognizing it, and of course, things became
Mhm.
more and more developed.
I didn't hear you for the last like 15 seconds.
Oh my, I'm so sorry. I don't know what's going on. Might be some interference. When you have a 2.4 gigahertz technology,
Mhm.
oftentimes even Wi-Fi may interfere with Bluetooth, and Bluetooth may interfere with Bluetooth. So usually I try to complement everything else around me, but we're surrounded by radio waves. They're not bad or they're not good, but oftentimes interference may occur, and yeah, it may be temporary or maybe more. And also in the sub-gigahertz part of the spectrum, a lot of industrial and a lot of government institutions use that part of the spectrum, and also pagers became increasingly popular amongst a certain part of the populace again. You may have heard of it that some of Hamas's leaders were eliminated using pagers, which
Hmm.
they bought at a premium price from a Hungarian company. And then at a particularly given time, it was game over for them. Mm.
Hmm.
Smart move. Usually, RNA carries transient information. After transfer, it needs to deteriorate. And is it double-stranded? Typically, it's not double-stranded. I mean, it can have loops if you remember tRNA, but yeah. Um, the OH of ribose can attack the adjacent phosphate, promoting a phosphodiester bond cleavage. That is why there is no deoxyribose in DNA. DNA, that is why ribose is not present in DNA. That is why deoxyribose makes up DNA, because the adjacent OH, well, it's going to be like phosphate. So stability is going to be a tad bit more hindered than in the other case. Yeah, so you can give reasoning to people why why this and why not that. Now, obviously, in many cases, in the phosphodiester bond, it's just a representation. 5 and 3 OHs basically grab a phosphate and they make a long, long, long chain. You want to see it here. It's kind of like that. It should be at the corner, but yeah, it's just a representation like in a school book. Adenine, guanine, cytosine, and thymine, they're going to be found in DNA, and they're going to complement one another. Obviously, through the hydrogens and oxygens, they're going to form hydrogen bonds. We're going to talk about them in greater chemical detail. I don't want to burden you too much with it. And they're going to form hydrogen bonds. Hydrogen bonds play a very important role in keeping the two strands together, but also stabilizing the strands is pi stacking. This is oftentimes ignored. Pi
Hmm.
stacking may occur through these aromatic parts over here. So the remarkable stability of DNA comes from double-strandedness, hydrogen bonding, the lack of an OH group. Yeah, but in deoxyribose, the deoxy part is important because it doesn't allow the cleavage of the phosphodiester bond. And RNA also needs to be mRNA especially needs to be temporary enough so it gets the job done, but it doesn't create a mess. And nobody wants to spend extra energy basically picking mRNA apart. It should do it on its own. It's ingenious engineering at its finest. If we were able to make a factory or a refinery or anything of the sort similar to a cell, it would produce a lot of profit. It wouldn't produce a lot of pollutants. It would be highly efficient. Sometimes adenine and thymine they're going to power up, and cytosine with guanine. In the case of adenine and thymine, two hydrogen bonds are going to be possible. The obviously the electron-rich participant in every case being nitrogen, and the electron-poor part of the other molecule being the hydrogen that is sticking out. And for repair, we don't need both strands. One strand is enough because the other side is going to be complemented. Not complemented like the guy from the Greeters Guild. You may have seen a mustached man on Instagram who greets people and says nice things to them. I don't know if he comes up to everyone. He usually wears a purple evening smoking jacket, and he looks like an absolute gentleman. And he says he's from the Guilders, he's from the Greeters Guild, and just says random nice things to people. In some places, he's being sent away, especially from stores, because it seems odd. And he's actually good for business, and he's just overall nice. And actually, he's doing this for fun as a publicity stunt. And he says he represents Jan Mosby. That man actually gave a goal pass to someone at a football match, and giving a goal pass is the nicest compliment. Yes, I mean allowing someone else to score is quite a compliment. So yeah, it's British humor at its finest, if you ask me. It's not that kind of compliment. It's basically adenine complementing thymine, thymine adenine, or guanine, cytosine or cytosine guanine. And when it gets repaired, it's basically being patched up with its proper opposite that's missing. Yes, in the 90s and the early 2000s, we used to have trousers that you could just zip the um, you can just zip the sides off and you can make them into shorts. It's kind of like that. If you can extend them back, then it sticks and basically it complements it. So when one part of your Lego building is missing, you can rebuild it just by looking at the other side
Mhm.
because it has to be symmetrical, basically. Now, what stabilizes the double helix? Of course, hydrogen bonding. It can be between oxygen and nitrogen as well, because there's an oxygen double bond as well, not just nitrogen and hydrogen. That is true. Base stacking is pi-pi interactions, and something we don't really talk about in high school is certain cations, sodium, magnesium. They shield the negative phosphate charges and stabilize the helix. So this is why it's so important to not lose sodium levels and also not deplete your magnesium levels. I usually keep little satchels of potassium and magnesium on my person, but don't keep them for too long. They can puff up, and if they do, that means something decomposed. Throw them away and use a new one. Uh, yeah, I opened one of those. Big mistake. Oh, by the way, today is the day when I'm going to buy one of those whitening pens that you've told me about.
Okay. How come?
Because I usually wear a white shirt and a bow tie inside with a three-piece suit and something. And yesterday we went to have Chinese food, and guess who was clumsy? A hint, not my wife. No, but the Chinese had some kind of thing there that basically removed it immediately. Imagine a place that is a hole in the wall and there's a kid writing his homework. I mean,
Uh-huh.
if it's a Chinese place, you're in for a delight, even though it's not the authentic, some kind of regional, but the Budapest Chinese, but good quality food. Yeah. And of course, guanine and cytosine-rich regions are going to be more stable than adenine and thymine-rich regions. What do you think the reason for that is?
Mm, there's more rings? No?
There's more,
Isn't
there's
it
more hydrogen bonding.
hydrogen bond?
Yeah,
Okay.
because one can do a double hydrogen bond, the other one a triple one.
Yes.
The
Yeah, that's
more
what I meant.
hydrogen bonds I have, the stronger the interaction is.
Mm-hmm.
Yeah, yeah. How do we make up nucleosomes? You already answered it before I would ask. It's
Mm-hmm.
just a revision. In many places, what I really love about working with you is that it's just revision. Yeah. Histons form an octamer, octa meaning 8, mer means units, or if you ever played the game Skyrim, mer means person, man, but that's a whole different story.
Hmm.
And as it twists around, it forms a nucleosome core particle, and as it rolls up through linker DNA, um, it's going to connect them as if they were beads. And if you ever made bead bracelets, you can twist them and compact them even more, and they're going to turn into a dense little ball.
Hmm.
We started doing that with the little guy at the age of 2. He loved it. It's always fun. It's always fun, and it teaches you about how 3D structures form. Because through playing, you learn, you imagine.
Hmm.
And with kids these days, what I have seen is that logic predates language.
Hmm. Okay, yes.
That's amazing. I find that remarkable. But I think we are studying children even more than we did previously, because, yeah, I mean, my dad's generation never really changed a diaper. Your dad's generation and mine, quite different in that regard.
Hmm.
The other thing is, a man will sleep through a baby crying, but as soon as something starts moving, they wake up and want to smash it just to make sure the family's safe. And that's perfect because that's how we're wired. A child needs a mother to nurture him or her, and needs a father to protect him or her. Yeah.
Hmm.
Again, nature doing its bit. Why does this matter? Well, because you really have to wrap up a lot of data. And you have to regulate accessibility. Certain parts will be unrolled, the parts that you need, while other parts are going to be more compact, the parts you don't need or you don't want to access.
Hmm.
And this organization compacts the whole thing for transcription, for replication, or even for DNA repair.
Hmm.
Well, because these molecules are rather fragile. Yes. Now, chromatin state and chromosome organization across the cell cycle, of course, changes. Cells have their own cycles. They form, they grow, they mature, and afterwards, of course, they're going to reproduce asexually, mainly while you're growing, or sexually when gametes form and they fuse. We're going to talk about that, mitosis and meiosis as well, because we need to master those. What I did back in the day, I would make my own silly little drawings with a pencil. I really sucked at drawing, but the more I practiced, the better I become at it. So
Hmm.
obviously, I've hit the ceiling quite early, but I do have friends who are very good at drawing because they have a talent and also they practiced more.
Hmm.
They were more inclined, but everyone can achieve a certain level of proficiency. And drawing cells and drawing basic little sausage shapes is really not that demanding. Oftentimes I would hear from, especially from male colleagues, I'm not going to draw, I'm not that good at drawing. Yeah, but you can draw a circle or a sausage. Come on. You just put the shapes together and you can draw what you've seen under a microscope. Of course, you can also photograph it. That's true. At this day and age, it's really not that difficult. However, drawing does other things as well. Or you can have AI generate things. Yes, that's true. It's far more visual, and then I'm all for it. I'm not against it, but. Um, Yeah, I mean, I remember I had colleagues who drew structures into their own books, and it was so uniquely them that it was really nice learning from them.
Hmm.
In the interface, it's less condensed, euchromatin is accessible, heterochromatin is compact. That's the difference between the two. Euchromatin is accessible, it's beads on a string regions, they allow gene expression and replication. That's the active part. Heterochromatin, it's tighter regions, more condensed, more twisted. It's probably more silent from a transcriptional perspective. And what we don't realize is that a good chunk of DNA isn't ghost DNA or junk DNA, it's regulatory parts that we didn't understand. So,
Mhm.
yes, oftentimes as researchers, we fall into the Robinson Crusoe mistake. We meet someone from a different culture for the first time and name them after the day of the week we've met them. So creative.
Mhm.
Now, post-DNA replication, replicated chromosomes and sister chromatids are joined. They're joined at a centromere. It's highly compacted. The term chromosome means colorful body,
Mhm.
somatic or somatic means of the body, so chromosome means colored body.
Mhm.
Chromosomes were perfectly unknown until we found proper poisons, basically, to freeze cells in the S or in the G2 phase or in the metaphase in mitosis to be able to see them. And we also needed dyes to make them visible. And we also needed good microscopes to be able to study them. So it's been a long road. A centromere is a constriction site that assembles the kinetochore that attaches to the spindle of microtubules. We're going to talk about mitosis a tad bit later. Telomeres are protective little caps at the end of the chromosome because those parts are fragile. We don't want them to break, so we add parts that just protect them. Here it's in dark teal. Teal is a reoccurring color. If you have a really old person giving a PowerPoint presentation, its background color is going to be teal. And you're
Mhm.
in for a treat because you're going to see research from 20 years ago presented as if it was cutting edge at this time. Cohesion is cohesion is a protein complex that holds sister chromatids together. You need cohesion to hold them together.
Mhm.
Now, obviously, this is the most condensed state of genetic data. Why? Because it's been duplicated and it needs to be pulled apart into sisters, into daughter cells.
Mhm.
Because the mother cell divides into two through mitosis, and you need to keep the chromosomes, the same number of chromosomes in both cells. And certain genetic diseases, for example, in the case of meiosis, occur because of the incomplete separation. Now, the weird part is going to come up now. This might not be that well covered in the IB program, but it's not your fault. It's not your teacher's fault. It's how the program is being made up. I mean, I read a few papers from the Royal Chemical Society just recently that should they reduce the amount that kids need to study for A levels? And I was at awe. I mean, they're already getting A pluses. 40% of applicants get an A plus. In the 90s, that was less than 1%. And
Mhm.
universities demand these extremely inflated grades, which basically undermines the whole role of education. Knowledge will suffer, and basically teachers are going to train kids for exams and nothing more.
Mhm.
That's unwise because in university they will have higher dropout rates, or universities will have to drop their requirements as well. And dropping demands, dropping requirements basically bastardizes education. Any kind of discrimination may be bad. You have to accept excellence and excellence only. So a
Mhm.
lot of British schools have brought about entrance exams again. So
Mhm.
your A level or O level will not get you in anymore, which I think is the healthy thing to do, because a lot of these degrees have lost a great deal of their value. With IB, I haven't seen such a pattern yet. What I have seen is the way that the material is packaged has changed, but the requirements were not dropped lower at all, which I think is overall rather beneficial, not just for the student, but for the community as a whole. Now, how a chromosome contains basically many genes and how these regulatory regions change what is being expressed. That is what we're going to talk about. Chromosomes have segments and a lot of genes and a lot of regulatory regions. And oftentimes these regulatory regions were called junk DNA or ghost DNA, despite the fact that nobody said that they were useless. You take them out of an organism and it just doesn't work anymore. And that's why they called it that because they didn't know what it did, but without it things didn't work. A gene occupies a certain locus within a chromosome DNA, and alleles that alternative sequences may be on the same locus virtually. And one gene may be promoted at a certain area and it will be more expressed because of an outside effect. This is how organisms react to outside effects at a molecular level. I mean, a single-celled organism cannot really talk. It will react with behavior and metabolism to the outside world. Usually, these single cell organisms are going to react through changes in their metabolism. Semi-conservative DNA replication means that one parent duplex is going to form two daughter duplexes, and one of these strands will be inherited from the mother cell. How
Mhm.
do we know? We know this from labeling. The mother cells were labeled, and the daughter cells always had 50% of their mother cells labeled DNAs, but just 50%. The other half was freshly made. There's a leading strand and a lagging strand when replication occurs, when there's a replication bubble. Sorry,
Mhm.
you were saying?
No, I didn't say anything.
Oh, you're just you're just making British noises.
No, I just probably just said like something like mm-hmm.
Okay, no, it's fine. No, whenever we're at a conference, we oftentimes make British noises, and people are like, your question. No, I'm just agreeing with you. Okazaki fragments are basically on the lagging strand, where synthesis of DNA isn't continuous. It occurs in these little pieces that are joined together afterwards. And the fork moves in both directions, obviously, and in this way, it's going to be far more effective and fidelity also remains true. Now, obviously, independent lines are evidence from the uh, 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 Stahl experiment. Basically, what happened there is they have labeled the mother cell's DNA and the daughter cells had half and half of the labeling material and bottom line that is how they found out that yes it is a semi-conservative replication. Basically nothing goes to waste and the new daughter cells don't have a copy of the mother cell. They have half of the mother cell and the other half is just being complemented, and that's about it. Homologous chromosomes, they come from maternal and paternal origins. And when we're going to talk about genetic diseases, we're just going to touch on them in general, and certain ones are going to be expanded a little more following Solomon's. But um, again, I don't want to burden you with too much of these because it can be rather um, in the realm of the Mangala show. Uh, my wife usually says when I'm teaching genetic diseases in pathophysiology, are you teaching the Mangala show again? Because it's just awful how many genetic diseases people can have, and yeah, it's creepy as hell. However, we have to be aware and we have to know how to avoid them. I'm also going to tell you about novel technologies that don't just help you avoid them, they can fix kids in the womb. For example, around 9 months ago, a child underwent heart surgery in the womb. They
Hmm.
fixed his heart and he was born without a scar.
Oh,
He's
okay.
perfect. He's perfect. He's healthy. It was a developmental, not a genetic issue. Everyone is well. It doesn't even show. If we would have had this technology 20 years ago, uh, Arbuston would still be alive. So,
Hmm.
yeah, whenever you're doing research, keep doing it. Keep doing it. You never know when you're going to save a life with it.
Yes.
So keep going, keep going. And it requires a lot of drive and it requires a lot of ability to cope with constant failure. It's really close to madness, isn't it? Doing something without results and yet expecting things to change. But they do. Ultimately, they do. You just need to figure it out. Yeah, people laugh or cry bitterly about it, but I have seen miracles happen in laboratories. I had a PhD student. He was working constantly, never had any results. And in about 2 weeks, he made 4 articles worth of substances because all of a sudden he figured out how to do that particular kind of chemistry because he tried every way that doesn't work. Now
Hmm.
he became a leading scientist at Richter. Then he moved to Switzerland because Richter didn't appreciate him that much, and he found a better job there. And he's leading a research group now. And Benedek is an insanely smart kid, and I'm very proud of him. But it took him a lot of trial and error, and he learned from it. This is what my PhD supervisor said as well. She had a colleague who got results just by being lazy, and he finished his PhD research in a couple of months and published in high-ranking journals because he was lucky he got an easy topic. My supervisor got the most difficult topic imaginable. Why? Because her supervisor imagined she's the only one who can do it. He was right, but she only published low-ranking papers and she was quite pissed about it. Yeah, but my supervisor became the dean of our faculty. My supervisor got the top prizes in chemistry in the country. My supervisor is asked by everyone for her insight in research, education, and diplomacy. I don't know what became of her fellow PhD student because nobody actually knows who he is. So even if we have difficulties, it doesn't mean that they're there to hinder us. Yeah, in religious terms, usually they say God breaks you before he makes you. Um, I don't know. I mean, I don't think that difficulties break you. They just help you cope more effectively. And with difficulties, we grow. Now, why am I saying this? Because medical school is full of things like that. And we're driven in medical school. And once you have a goal in sight, you're moving towards that. And you can move a mountain with your will alone. I've seen that with my sister. People are generally afraid of her because she's really strong-willed. She finds a purpose, she finds a goal, and she executes it.
That's great.
Mm, yes, there's no other way. And I've seen that with my colleagues as well, who are medical doctors. Once they set out a goal, I'm going to tackle that problem and make it vanish from humanity. They're going to do it. Yeah. And usually they say the weaker sex and how fragile women are. You know what? I would rather be locked in the room with a polar bear and a grizzly bear than my sister, and them, of course, attacking me. Of course, I'm exaggerating a little bit, but yes, I do respect this kind of determination towards development. And once you know you're on the right path, you're gonna keep moving forward.
Hmm.
The chromatids are replicated copies of one chromosome joined at the centromere. So they're going to be popped apart and they will appear in daughter cells. From homologous chromosomes, one came from mom, one came from dad. They're in the same cell. Sister chromatids are going to separate into two sets and become a new somatic cell.
Hmm.
These are oftentimes confused by kids. That's where I'm going with this. Now, diploid cells have two chromosome sets. Haploid gametes only have one set. We humans, as our somatic cells are diploid, and our gametes are haploid. Is this true for the whole for every living being, especially plants? Not
No,
at all.
no.
No. Cavendish bananas, they have this set of chromosomes. They're triploids. Why did Cavendish bananas come along? Why do they even exist?
What kind of bananas?
Cavendish. Cavendish, like the encyclopedia. Cavendish.
I'm not sure.
There was a white bananas were always triploids.
Hmm.
And way until the 1950s, bananas tasted like fake banana flavoring tastes like right now. And then a powerful disease hit them, and since they were a homoculture, they were not reproduced via sexual reproduction because, as you can see, bananas have no seeds. They have those black little dots, but they never form into seeds
Hmm.
because they have been bred out of sexual reproduction, and they're usually vegetatively reproduced, just like potatoes.
Hmm.
And because of that, obviously, entire fields were becoming homocultures, monocultures, and when a disease hit, it decimated the plants. Cavendish bananas were a new strain that were more resistant to the disease that hit bananas. However, they were bigger, but flavor-wise, not as powerful as the previous ones.
Hmm.
So that's most of the bananas that you can buy nowadays are Cavendish bananas. They're good, they're full of fiber, they're healthy, there's nothing wrong with them. And you may have heard of the potato famine in Ireland when the potato blight hit Irish peasants.
Yes,
People
I think
died
so.
of starvation, and the country was exporting food while its people were starving. The British landowners at the time, they purposefully decimated the Irish and basically had them die of starvation. A lot of them left for America. Those were nasty times. Those were nasty times, and I kind of understand. But the issue was they had a monoculture of potatoes, and the main source of calories were also potatoes at the time. You'd be surprised to learn that people can live on potatoes alone. Yeah, I mean, obviously, if someone's doing hard manual labor, 6 kilos of potatoes a day will keep them going. And when you go to an Irish household around Christmas, you're going to find potatoes done at least 7 ways. And they're good at potatoes. It's just odd how much they love potatoes. Yeah, there was also jokes about people from the northern part of Kingdom of Hungary that they eat potatoes with bread, and that's why they lose their hearing. And it was a joke people would make about themselves, by the way. And yeah, it's not bad actually, to be honest. I mean, at home I have people who order a shawarma with fries and nothing else. And they have it wrapped up and they eat it like that. And I was surprised. How can you eat that? I mean, we make fun of the British for eating beans on toast, and then there's that. But it's not bad actually. It's pretty good. But it's starch with starch and yeah. Haploid cells contain half of that set of chromosomes. However, sexual reproduction produces genetic diversity. These babies being triploid and reproducing vegetatively means they're a clone of a clone of a clone,
Mm-hmm.
hence more susceptible to disease. Autosomes are your body's chromosomes. We have 22 pair of those and one pair of sex chromosomes. Ladies, they
Mm-hmm.
have two X's. Gentlemen, they have an X and Y, and I'm certain I have offended certain ideologues, but I am really not concerned about that because the truth is the truth. Now, one of these may become inactive. It may become a bar body. We're going to talk about that later on as well. This is why tricolored cats are usually female because their fur and their skin is patched a little bit. And this is true for every female. Obviously, it's not visible on a woman's skin. Maybe under a UV light, but not on everybody. But it's true. Our bodies deactivate one of the X chromosome. One is dormant, the other one remains active. This is why oftentimes women are more shielded from X chromosome linked mental health diseases as well. Oftentimes it's carried by the X chromosome. This is why I tell boys that if you don't have any mental health diseases, thank your mother. And
Mm-hmm.
it's true. And it's true. We're going to learn about this in greater detail later on. But of course, if chromosome 21 has a trisomy, it's called Down syndrome. Why a syndrome? Why not a disease? Because the level of damage and the interaction goes on a spectrum. Yeah, well, someone could have Down syndrome and still not be that much affected. Obviously, it's not a blessing. And in Japan, from since last month, there won't be any more Down syndrome babies born. Why do you think that is? Did they become Nazis and kill all of them?
No.
No, definitely not. I hope to God not, because nowadays when I'm just clicking on a news link, I'm like, is this a joke? Is this humor? Is this satire? Or is this reality? Because at this point, oftentimes I can't tell the level of insanity. No, they came up with a method to get rid of the extra chromosome and have perfectly healthy babies being born. Hats off to the Japs. I mean, obviously, you shouldn't drop two nuclear bombs on a nation, but yeah,
Uh,
they've done some amazing and some really weird things. But it's a huge and diverse group, and they have their own difficulties, but their researchers are remarkable. Bacterial chromosomes, completely different. Circular DNA, no telomeres. Why? Not necessary. Oftentimes, they've theorized that bacteria and anything with a circular DNA is biologically immortal. Unless it gets really, really damaged, it's going to live forever. And eukaryotes, because of telomeres breaking, might die earlier. Well, here's the joke. We have telomerase enzymes that repair our telomeres. So, no, not really. Not really. But for a while, that was the leading theory in science. For a while, till 10 years ago. And I'm not teaching the same thing I used to teach 10 years ago. I'm not teaching the things I've learned in university.
mhm.
Why? Because science, especially cell and molecular biology, is developing so much. A
Yes.
plasmid is a moving genetic piece of material, and obviously through plasmids we can introduce pieces of DNA. plasmids can replicate independently and via conjugation, because between you and me, just don't tell anyone else, yeah, bacteria, they can have sex.
Okay.
Well, not not in a pleasant way, but when they're really threatened, they're like, you know what? We're gonna die. Let's do this. And like, holy cow! When I found out, I was like, no way. And my teacher was, way. They can form a sex pilus and exchange plasmids. Why? In order to contribute to greater genetic diversity and kind of sort of praying to God that one of us is going to make it because we shuffled the cards. Yeah, because otherwise they're a copy of a copy of a copy. Eukaryote chromatin is a tad bit different. We got those pesky little telomeres, and we have histones, we have other proteins, highly organized, highly compact, highly regulated genome. If something is really complex, a lot of things can go wrong, hence cancer. In bacteria, something goes wrong, it's really simple. That's it, we're dead. But in eukaryotes, there's a lot of self-repair mechanisms as well. It's just like in the military, there are certain redundancies built in order to keep things going and keep them alive.
Mhm.
Have you ever wondered why doesn't an aeroplane have just one pilot? Why does it have a co-pilot?
In case something goes wrong.
Spot on. They don't even eat the same meal from the same lot
Hmm.
in order to make sure that everybody's safe. Now, what's the core logic? Nucleosomes are compact pieces of DNA, and they allow regulated access. Methylation, acylation decides that. And of course, this can come from the outside world or from inside the cell. One has to be careful to avoid alkylating agents and acylating agents because it can cause random mutations. Can you avoid them perfectly? Not in this day and age, but we're much healthier than we used to be. First of all, we're not that much exposed to smoke. A lot of East Asian ladies, they die early of lung cancer because being exposed to smoke due to cooking.
Hmm.
It's just
Oh.
insane.
Mm.
Yes, nobody talks about this. Have you noticed that there are very important topics that are much worse than what the media is regurgitating? And basically, if it's useful for certain influential circles, then they're on it. Otherwise, no. Human rights don't matter if the humans in question don't represent any kind of community that can be exploited for financial gain. Obviously, or for votes. But I think all human beings deserve a shot at everything.
Yes.
So an open and relaxed chromatin is more accessible, often more permissive. And as you can see, there's those AC acylation makes them more open, more methylation. On the other hand, any kind of alkylation says, no, I'm closed up. No. So these two mechanisms are both at play. These two have to be in proper equilibrium, proper dynamic equilibrium. Dynamic equilibrium is like a seesaw. Basically, it maintains itself with constant movement and whatnot, and that is a very important thing because certain things need to remain silent at sometimes, but they also need to remain accessible. Something goes wrong, and then certain types of cancer may appear, or with increased age, acylation, methylation may have issues. I have a paper on that in Hungarian. If you like, I can send it your way.
Mhm.
Euchromatin versus heterochromatin. Yeah, through aging, certain immune cells go crazy and they start developing all kinds of immunological cancers because they're older. It just it's just insane how these things occur. And obviously, if you're a lifelong smoker since the age of 12, you're in for a treat. You're in for a treat. Yeah. Ever since I could talk, I would ask my mother nicely to reduce the amount she smokes. Her dad died of smoking. Please don't do this.
Uh-huh.
Oh, the only person who was able to save his parents that I know of is your dad.
Yeah.
In fucking incredible human being. When I heard it, I was like, oh my God, it feels so good to see that, you know.
Yes, definitely.
Yeah, it's cool. Euchromatin is more open. Heterochromatin is more closed. Does that mean it's completely open and completely
No.
closed? No. Some things can be more accessible, others less accessible. And this pattern of accessibility is oftentimes a thing that influences cellular response.
Yes.
Chromosome territories and domains are oftentimes going to exist and inside the nucleus, and the nucleolus is a very important regulatory area. There are nuclear territories, and they organize DNA into functional domains that enable regulated genome function. Something breaks up, then we're in for a treat. Local looping brings elements together, and a loop domain is going to be quite interesting. And if you have access, that means it's more acylated. If you have lesser access, it means it's more alkylated.
Mhm.
Now, when chromosomes form, basically this occurs during cellular division. The centromere region supports kinetochore assembly, and a microtubule set will form, and the microtubules are going to basically rip the whole thing apart, and the sister chromatids are going to go into the new
Mhm.
cells, because there's going to be two of them. The telomere cap, as you can see, it's not nonsense. It's TTAGGG regions again and again and again, which is great, but you can have too much of the good thing. Yes?
Mhm.
With the telomere cap, it's more protected. They're not ordinary protein coding genes. They're not, they are protective chromosome regions. Can they be restored? Yes. Are they being restored? Definitely. Now, a telomere represents these regions, but there's also a protein cap called and sheltering proteins bind telomeric DNA and organize into a T-loop, and it has the chromosome end, and it prevents degradation, and it maintains genome stability.
Yes.
Yes. Now, in the S phase, obviously, replicated, you're going to have two sister chromatids, and after replication, they're going to be pulled apart. And in the N phase, sister chromatids are going to separate, and each daughter cell gets a copy of the same chromosome. Replication produces these sister chromatids that have to be properly segregated and accurately later on. Through semi-conservative replication, we get one strand from the mother cell, and the other one is obviously going to be freshly produced, and daughter cells get half of the original DNA of the mother cell. And oddly enough, in Hungarian, we also use the same term, mother and daughter. Yes. Yeah, I mean, if it's asexual reproduction, it's definitely girls in Hungarian. Otherwise, gender talk is not that common. It's like, "Ugh, amazing." But if you really hate him or her, it's like az ott me. Even worse. So there's going to be complement with the same thing. It's going to be templated synthesis. It's not just random things being popped to the other side. No, it's what fits exactly on that side. You get half of something and you're able to reconstruct the other half. Basically, that's what the semi-conservative thing means. Ah, yes, and the wonderful nitrogen isotope labeling experiment, basically half and half. One intermediate band after one generation is what has supported the semi-conservative replication,
Mhm.
the Maselson style. experiment. It was easy. It was conducted in a test tube, and that's that. Now, obviously, the replication bubble is going to form. Replication begins with specific origins and proceeds in both directions. A replication fork forms, and initiation proteins bind on one side. Helicase, basically, with the two strands apart. Zinc is absolutely necessary for the zinc fingers to pull apart the two sides of DNA. I'm showing it in the air despite the fact that my webcam's inactive. And obviously, helicase opens DNA to form the replication bubble. And for that, zinc is necessary. If zinc levels are low, obviously cells won't divide as much. And that's why after proper dehydration, first element you need to supplement with is going to be zinc.
Mhm.
But you already have practice in primary care of dehydration, I presume. So there's a left fork and a right fork, and there's the origin in the middle, the leading strand. It's going to synthesize DNA in the opposite direction in comparison to the lagging strand, which again DNA ligase is going to link together the Okazaki fragments. Topoisomerase is going to unravel the whole thing. Helicase is going to open up the two strands of DNA. And Okazaki fragments, well, they've been discovered probably when you were born. I think it's that recent. The replication bubble contains two forks. You have the leading strand and the lagging strand. And obviously, in the leading strand, DNA is continuously synthesized in the lagging strand in the form of Okazaki fragments, which are linked together. And topoisomerase unravels things, and helicase breaks up the double helix.
Mhm.
Now, the left arm is the leading strand. It's moving towards the fork from 5 prime to 3 prime. This has to do obviously with the sugar's direction because it's a polyester basically. Phosphate, sugar, phosphate. So yes, it's imagine as if little stickman figurines would be standing there holding each other's legs and with each other's hands through a phosphate. And the other leg is missing because it's deoxyribose, not ribose. Yeah, it's kind of morbid, but it does represent what's what. And the replication fork goes in both directions. Okazaki fragments occur in pieces, and then ligase and polymerase basically links them together, and the two strands are going to be perfect copies. Yes, lagging strand fragments are a consequence of chemistry and geometry. That was the best way the whole thing could be executed. In the
Mhm.
leading strand, basically, it's one continuous stretch. In the lagging strand, it's smaller bursts of synthesis, synthesized away from the replication fork, lagging strand, and leading strand towards the replication fork. One primer is enough for the leading strand. More primers are necessary for the Okazaki fragments to be added together. It's a bit more extensive, and it needs to remove gaps, but ultimately the two strands are going to be equal and perfect copies. Yes.
Mhm.
Now, the direction of the fork is easy, it's understandable. Single strand binding proteins are obviously going to be coated by the appropriate enzymes. ATP is definitely going to be necessary for it, and unwinding is going to need ATP. Stabilization is going to require ATP. It's going to require magnesium. It's going to require sodium and potassium.
Mhm.
Primase, basically, DNA polymerase cannot start on its own. It needs something to start things. It's basically temporary primers because DNA polymerase requires a spark. Well, this is the spark, then that starts the whole thing. And these are basically RNTPs. Polymerase extends from the primer in the appropriate direction. And it's not permanent. It's going to get discarded afterwards. You just need to begin somewhere.
Mhm.
After the whole thing occurs, what's necessary, just like when you create a PowerPoint, proofreading, proofreading.
Mhm.
Obviously, very small mistakes may remain, but we try to avoid them. And if everything's all right, then obviously we're gonna move forward. If it's not, cut out the bad part and insert the good part. There's no redo because that's
Mhm.
energetically much too demanding. Okazaki fragments, they're on the lagging strand, and basically on the lagging strand, fragments are processed in seals, close nicks, no gaps, for missing DNA, but only the parts that are not necessary are being cut out, and they need to complement perfectly the other strand. Um, yeah, ligase uses ATP or NAD depending on the type of organisms. Eukaryotes go more with ATP. Bacterias only may have NAD plus dependent ligases. NAD plus is oftentimes consumed as a supplement nowadays. I'd be careful with it. It's not well researched yet. Creatine is perfectly researched. However, with NAD plus, I would be much more careful because cancer cells can be able to have a much more effective metabolism because of it, and also germs and bacteria. It's not something we want. The replication fork is an absolute well-coordinated machine. Unwinding, stabilization, priming, elongation, maturation, and proofreading occurs. When everything's okay, we're fine. Let's compact it back into chromosomes. Let's split it up. Basically, that's the bottom line. Easy,
Mhm.
understandable,
Yes, definitely.
in a nutshell. I think it's fair right. Now, in the case of prokaryotes, it's a little bit different. Basically, circular chromosome, single origin. There's no end in a circle, is it? And
Mhm.
it's naked DNA. It's so easy. Seems like a walk in the park. Well, yeah, but imagine, I mean, the higher the complexity, the more things can go wrong, but the more refined results you can have. Well, this is the thing with prokaryotes and eukaryotes. Basically, we have linear chromosomes and linear DNA, many origins, linear ends with telomeres to protect them, and wrapped around histones, protected in every way, much higher stability, much higher control. And of course, one happens in the cytoplasm because prokaryotes, no nucleus, while the other one happens in the nucleus, protected from everybody else. So absolutely guarded. And the coping logic is concerned, but the origin number and chromosome structures have to differ because there's different levels of complexity. DNA synthesis is driven by adding deoxyribonucleosides triphosphate, which can release a phosphate, basically similar to ATP, not analogs of ATP, but it's DATP. Well, it's available, and the high energy phosphate releases energy the same way. It's basically cash. It's basically payments. Now, obviously, we need to have high enough speed and high enough accuracy. So compromise. If necessary, mismatch repair is going to occur after creation, and high fidelity replication ensures that we have a lesser likelihood for random mutations. Is it zero? It's never zero.
Mhm.
primers are going to be removed. The strand is going to mature. RNA primers are starters. Mature DNA requires a backbone continuity, and when it's removed, hydrogen bonds reform, and basically DNA ligase gets rid of nicks and repairs the whole thing. Now, RNA, there's a few misconceptions that they're all removed and replaced with DNA. Basically, what happens is that the RNA primer is a short RNA made by a primase. The nick is some kind of break. When you nick something, it's used mainly in the British vocabulary. It might
Uh-huh.
be a break on one side, but it can be mended. Yeah, I nicked my knackers. That means I ripped my trousers or my undergarments, yeah? And someone's going to mend it. Mend it means fixing it. Oftentimes it meant sewing
Yes,
something. It's from slang. I'm not sure if this kind of vocabulary is still used in England at the time. I guess it depends on the region. DNA ligase seals these nicks, and the strand matures and it becomes whole again after the two parts have been resealed. Now, topological stress has to be managed. In order to avoid it, you cut it and reseal it because if it's too tight, it may break. Imagine twisting beads, and you can twist them until you break the strand somewhere. Have you ever played around with such things?
definitely.
Yeah, I mean every kid does, but I don't think you went as far to break it. That yeah,
Oh,
you can feel the emerging frustration. Well, it's trying to diffuse that. Topoisomerase 1 and topoisomerase 2 can cut. Topoisomerase 1 can cut one strand to reduce stress and then reconnect it. Topoisomerase 2 can cut both, and basically it's trying to reduce the level of frustration of the whole thing. Topoisomerase 2 is oftentimes ATP dependent, but while 1 is not that much ATP dependent, it can use other source of energy likewise. The telomer and basically primer removal at all the chromosomes may create a shortening problem in linear DNA. However, telomerase fixes that. It ends those ends, and this is why it was theorized that telomere is the candle of our lives, and if it runs out, our cells die. To an extent, yes, but we have mechanisms to repair that. So basically, you may know this from folklore that there are, I don't know, some kind of mythical beings there that are burning the candles of everyone's lives, and you just go there, bribe them, and have them add a longer candle to your life, and you're all set. These things do occur in folklore, and it does use the Dr. Faust theme, but in Central and Eastern Europe for some reason. Well, in real life, they theorize that telomere is something like that. To an extent, yes, but not really. Because we have telomerase enzyme, and telomerase, well, it can do a number of lovely things. First of all, it can repair this telomere end. So, yeah, it's very active in germline cells, adult stem cells, and in a number of cancers, unfortunately. So, yes, in cardiac muscle, erythrocyte neurons, it's not as active. Fun fact, when you're pregnant with a child and you have cardiac muscle issues, the fetus will do everything in its power to save you. It will send its own little stem cells and repair your heart, and you're going to survive it. Same thing applies for other diseases. So, anyone who's ever been pregnant with a child bears a mark of each and every pregnancy in the form of these dormant stem cells. It's a double edged sword. They can contribute to immunity, or they can interfere with the immune system and become cancers. It's called microchimera, the
Mhm.
magyarul mikrokiméra. It's basically same species but two different types of DNA, and the mother's organism usually doesn't refuse the child's stem cells. So theoretically and even practically, if you have, for example, your mother underwent radiation therapy, chemotherapy, her tissues aren't able to produce proper blood cells, proper immunity, or her stem cells are worn out, you can just pretty much harvest your own stem cells, and a couple of milliliters of it can be injected into someone's bloodstream, and they will find their way in the other person's body and start growing their own little colonies and reforming its blood producing stem cells and also damaged tissues. In some rare occasions, they're also able to encircle cancers in the mother's body and then go into apoptosis and start accumulating calcium and killing the cancer cells selectively. Yeah, it's remarkable what stem cells are capable of, and in China they are extensively researched. Mind you, they're not magic, but they can be really useful. And I have indeed seen nothing short of a miracle being done by them. However, if you're an adult and you decide to donate some of your stem cells in a couple of months, your other stem cells might go crazy and you might experience um, a partial whitening, for example, of your hair.
Hmm.
Otherwise, no other real effect. But it can be indeed a lifesaver. However, through medical ethics and European law, you cannot force anyone to undergo therapy if they don't want to. That's a very important thing. In other places, it's different, and on international waters as well. So you cannot save anyone against their will, and that's extremely important because you can get into more trouble than you're set out for. Now, telomerase again is a double-edged thing. It's inactive in muscles, however, telomerase reactivation occurs in cancers because they start acting as if they were stem cells and they start reproducing, they go into metastasis. Not all cancers are the same. That's another killer thing because a lot of cancers vary. You can have a lump somewhere and it may contain thousands of types of cancer cells, and you may be able to eliminate the majority of the lump by selective chemotherapy, and the ones you weren't able to target may start growing instead. So it's a really difficult battle, and there's no universal silver bullet against cancers. Calling it cancer is kind of outdated, but there's no better word for it. And the issue is that one tumor can be really complex. If you manage to target the outside and cause calcification and force it to destroy itself, and the inside doesn't get proper nutrition, then you might have a winning chance. Yeah. Now, shortening may put a pressure on tumor persistence. However, your own germline cells can suffer, and also your own quickly regenerating tissues. For example, if your hair grows back really quickly, that means your stem cells are extremely active. If your toenails grow really quickly, that means your stem cells are really active. In the case of men, if you have toe hairs growing, that means, yeah, you're doing well, you're still alive.
Co-hairs.
So yeah, I mean, in the case of Hobbit and myself, obviously, but yes, yes, I've seen it on a lot of people in the countryside as well. And since I turned 40, for shame. But yes, yes, and it's a sign of your body still being able to regenerate and still being able to grow things. And if your wounds heal remarkably quickly, that's also a good sign. And you have to maintain that with proper sleep, proper rest. This is all coming from a guy who used to sleep 6 hours, but now I manage to average 8 hours with sleep maxing. So yeah. And of course, replication stress is unresolved. These checkpoints are being lost. Then cancers are just gonna keep growing and growing and growing and consuming. If damage occurs, that's one thing. Replication error is another. Damage occurs, then of course the bad version is gonna be copied over and over again. You can have UV radiation alteration. It can occur anytime. What is the greatest and most of an unavoidable carcinogen on the planet?
What's the biggest carcinogen on the planet?
That is unavoidable.
Radiation?
True,
No.
but
Sun?
electromagnetic radiation,
Oh.
light, the sunlight, yes, it's what fuels us. But again, it depends on which part of the spectrum because our biggest organ is our skin. Will it kill us? No. Can it damage us? Yes, in excess, and it can cause damage. Yes. Are we going to die of it? No, because we're careful about our skin and we can take good care of ourselves. If there's a mismatch repair, it's going to create bubbles. It may trigger apoptosis. Bottom line. Base excision may occur as well. A base gets cut out. It's going to be patched up. It's going to be filled up. A nucleotide excision may also occur. This one shouldn't be here because it's not on the other side. Let's cut it out and patch it up. Obviously, in Xenoderma pigmentosum, it's an extreme sensitivity to UV light. And these kids, we had a set of twins. They were only allowed to go out at night. They were particularly white. And they didn't live for long, unfortunately. They died in their teens. Um, the whole community said, "Oh, they are sick," and because they could only go out at night because of this condition, they thought they were in cahoots with some kind of dark forces. No, they were just sick. The double strand may also break up. This is just a few cases of what happens when things go wrong. Obviously, mutations may also occur. You may have one nucleotide change, small parts change, some things may be repressed, some things may be reactivated, and it depends on the level of mutations and what's going to be the consequences. You can have silent mutations that are not apparent, missense, nonsense, and frameshift mutations. Was the difference. In the case of a silent mutation, there's no effects. A missense alters one amino acid, and it may cause disease like sickle cell anemia, changes one amino acid. Nonsense, non-functional due to premature termination, it's going to be severe. And frame shift, it can be severe because it's going to shorten the protein and it may become ineffective. Somatic germline mutations. Somatic mutations of certain somatic cells are not going to be inherited, but germline mutations are going to be inherited. If you've seen the film Chernobyl, the boys who went there, they were told to wear lead kind of sort of underpants or loincloths. And when these miners were talking to one another, they explained to the younger miner who just managed to grow his first mustache that wear this or your children are going to be mutated or you're going to get sick now. One looked at the other, wear this or your thing is going to turn into a vagina. That was it. And he was like, okay, I'm wearing it. But yeah, they were quite heroic. I mean, they made the ultimate sacrifice in order to protect everybody else because some higher up made a stupid mistake. But yeah, these miners were absolute heroes and yeah. A lot of them died of radiation poisoning, and a lot of them couldn't have healthy children. So, yes, the entire lineage is going to be affected by these mutations. But that's just one thing, radiation. When the US bombed Vietnam, it used Agent Orange, which caused mutations in the local population and also in the offspring of Vietnam War veterans. Yeah, not in the first generation. The second one, grandchildren were born with limbs that didn't work and things like that. Yeah, this is just awful. How can you prevent mutations? Well, there are internal mechanisms as well. Obviously, use proper sunscreen, don't expose yourself to nonsense, but sometimes you just can't. But obviously you can stack up on the good things. Yep. Uh, Proper sleep, proper rest, try not to worry, grow a set of P53 genes, at least 8 of them, like elephants. Have you ever seen an elephant with a cancer?
I was in what?
An elephant with a cancer.
Elephant.
Usually you don't.
I don't think
They
no.
no, they have 8 copies of the P53 gene, and it's very rare for an elephant to produce cancer, despite the fact that they're big and they live long. So yes, it's a complex thing. Telomeres have to be properly maintained and make sure that genes are copied properly. Obviously, in a demanding environment, these can change. However, the way you feel about yourself, your nervous system influences your hormone system, which influences your gut microbiome. So it's a complex thing. UV radiation, alkylating agents, intercalating agents, oxidative agents, they can all damage DNA. These things need to be avoided. Repair responses are there. Um, the damage may be done, but it can be avoided. Intercalating agents can be dyes, antiseptics, lab reagents. Cisplatin, carboplatin, for example, are used as anti-cancer agents, despite the fact that they're intercalating agents. Alkylating agents, obviously, they exist in great amount. Basically, alkylating the bases deactivates them, and UV radiation isn't necessarily something we can avoid, but we can avoid it. I mean, you don't have to expose yourself to a lot of UV radiation, and also you need that vitamin D through tanning, obviously. There are a number of proofreading checkpoints and cell cycle checkpoints that make sure your cells are going to be healthy. Chromatin after replication is going to be reformed. Everything's going to be reformed into sister chromatids. They're going to be pulled apart, and everybody gets a nice little copy of it. And this has to do, obviously, with the cell cycle. And if everything in its place, if everything is properly coordinated, you won't have any issues whatsoever.
Mhm.
Mitochondrial DNA can pose a threat and a danger. It is usually from both parents, but mainly from the mother. And daughters and sons can inherit them equally. So it's not going to be like I'm the one who's going to inherit something because I got it from mom. And of course, nuclear DNA is biparental, mitochondrial from the mom. Plasmids as extrachromosomal DNAs, as I said, they can contribute to the survival of bacteria under extreme conditions. They're called moving genetic materials. Antibiotic resistance, virulence factors, metabolic advantages, and stress resistance may be exchanged so the species itself survives. So if something effectively damaging comes around, otherwise asexually reproducing bacteria start growing tecpili and basically exchanging these factors so they have a better chance at survival. Genes are not proteins, nothing new here. Yes, it has been a concept for a while. Alleles are variants, not necessarily deterministic. Later on, we will talk about these in greater details, that there are variants at a certain locus, and context matters very much. Regulatory bridges also occur. Obviously, this is way above what's necessary. And of course, certain proteins read DNA effectively, transcription factors and repair proteins, in order to make sure it remains intact. Proteins recognize too shapes, grooves, base exposure patterns. Oh my, it's only 27 degrees in Budapest. It's rather pleasant.
Huh. Okay.
Yeah. And again, it's again that bit of a recap. What happens when something works? What happens if it doesn't? Again, a different approach to the same thing. These further slides are just going to help you understand what we already covered from a different approach, from more of a what happens when things go wrong kind of approach. If telomerase isn't active, there's going to be issues. It's not an immortality enzyme. It was widely sold and marketed as such. Unfortunately, people can be easily duped, and you would think, oh, they're probably scientifically illiterate. No, they actually claim to read a lot, just selectively reading what supports their ideas. I had a flatmate like this, Patrick. Patrick would always read up on things. He would have dimethyl sulfoxide in his room, 1 liter of it, owning 1 milliliter of dimethyl sulfoxide as a private individual lands you in jail for 1 year.
Hmm.
Patrick, my man, he could probably be jailed for 1,000 years. Nobody cares, obviously. I mean, uh, but he kept injecting himself with random stuff, and then he would have random weird issues because of it. Self-medication, even if you're sick, is a bad idea. Proper analysis and of course, proper medical assessment is necessary. And you know, it bothers me because I care for the man, because he's a genius. I mean, he can repair things just by touching them, feeling them up, and he has an aptitude for this. But he didn't have the stamina to earn a degree in engineering. So he fixes random things, and he's a maintenance guy at a university, and he lives in a dorm. It just sucks.
Hmm.
Aneuploidity and karyotype limits may occur, as you can see, if there's too many or less of certain chromosomes. It can mean a genetic disease. Is it always a genetic disease? No, it depends what is being affected. The trisomy of chromosome 21, it can be a huge issue. 47 chromosomes, having extra X chromosomes, can it be an issue? Yes. Having two Y chromosomes, can it be an issue? Yes, but not that big of an issue. I have known 47 chromosome men who have two Y chromosomes. They're taller, they're a bit manlier, they're not more aggressive, has nothing to do with criminality. They may have slightly elevated levels of testosterone. They may develop speech and reading skills later, but they're not dumb and they're not criminally insane. There was a paper published in the 70s, which obviously doesn't make sense because it was bad science. And I know of quite a few cases in Budapest that women terminated their pregnancies because through extensive genetic testing in the 80s, they found out that they're expecting a son who has two Y chromosomes.
Hmm.
And the little guy would have turned out amazing, but he never made it. So I think it's a
Oh.
tragedy.
Hmm.
I mean, don't get me wrong, I understand a lot of things and whatever, but life is something that should be cherished, and every life is a
Hmm.
gift, and a chance is something amazing. There are a couple of misconceptions, but these are basically only study sites, so I don't want to keep you for longer if you don't mind. But if any questions come up, then of course